Good morning, everyone. I hope I am visible and audible to everyone. I am not able to hear anyone. If anyone could please tell me if there is a sound issue.
Now you can talk. Hi, I can hear you now.
All right. Thank you, sir.
Can you hear me? This is Dr. Shivakumar.
Yes, sir. Good morning. I can hear you very well. Thank you.
Right next to me is Miss Jennifer.
Right.
Good, we are on.
All right, sir. So, good morning, everyone, and welcome to the third day of Equinox 2023, an international conference that has been organized under the aegis of the Center for Research, Maitrey College, University of Delhi. We are on the final day of this conference, and today we are having technical session 7, where it gives me great pleasure to welcome our two esteemed guests for the session, Dr. S.S. Shivakumar and Ms. Jennifer Pandian. So without further ado, let me first give you a brief introduction about these two very esteemed guests. Dr. S.S. Shivakumar is one of the global pioneers in manufacturing and marketing of AME machines for about 2 decades, and he's the first ever patent holder for AME process filed in 2004. Sir has been a consultant for World Bank, the Ford Foundation, and the Government of India for water resource management. He's a man of many missions and has over 40 years of distinguished experience both as a researcher and an academician, a serial entrepreneur, philanthropist, and is currently heading the research division of Thales Cleantech. We extend an extremely warm welcome to you, sir. Thank you for coming. Ms. Jennifer Pandian has 10 plus years of professional experience in the water energy nexus domain and has been actively involved in research and product development of AME technology since 2012. Ma'am has hands-on organic experience from implementing AME technology in various parts of the country. As per her academics, she is an MBA graduate from Germany with special focus on international cooperation and sustainability, is a certified water professional by the World Bank and the United Nations, and a strong believer in trusting the process. We extend a hearty welcome to you, ma'am. Today, we are indeed privileged to have a talk with both these very distinguished people on a burning issue of today's times, that is on water management. So I am very happy to get an opportunity to handle and moderate this session, and invite both our distinguished speakers for today's session, which is the title is um, Innovation in Water, How They Change the Questions in Social Transformation. So over to both of you. Thank you.
Thank you very much. And a happy morning to the chairperson, to the other participants, and all the students present. Good morning to you all. Um, we're talking of social change. The biggest social change is that which happens across civilizations. So, the first ever major civilizational change happened when the pastoral civilizations of this world found the art of how to manage water. So, water management was the first major innovation that led to transformation of society in a fundamental way. Starting from that, we see a long history of humanity dealing with water. The interesting question is whether water was so central to humanity that they needed it to survive. Yes, indeed, because all the major civilizations in the world have been located near water sources. Having said that again, I'd like to just bring you update to where we are now. We are talking of the year 1924, 2024, and we are talking of the year when we began our first experiments with making water from air. This question arose because we had become aware that India, with about 16 to 17% of the world's population, had only 4% of potable water reserves of the world, which is really a critical situation. And most of this 4% was heavily contaminated with heavy metals, arsenic, and even biological contaminants. So we were at a point where we were not looking at water management because there wasn't much water to manage when we were looking around. We were thinking of trying to find water from some source which was unknown till date, not any groundwater, not any surface water. We had to look elsewhere. So in 2004, we started looking at the atmosphere because the largest storehouse of water in this planet is the atmosphere. Approximately 500,000 kilometers of evaporation happens from the oceans of this world, and which is all in the atmosphere. The idea was that we should tap it and finding a technology that would tap this water for suitable human use. This is how we began. And now I'll let Jennifer take over from me and tell you how we began and we went on.
So when we began in 2004, we started making these missions. Those were the times the packaged water itself is not very familiar in the market. It's very few, institutions or companies, they use bottled water, especially when they have conference or meetings. People were not used to the concept of drinking water in the packaged format. And that time we introduced something, uh, very ahead of their imagination, that there is a mission which gives you water from the air. And uh, yeah, we somehow got our breakthrough when we work with the government, defense, and Ministry of Health. They were very open to experiment with our technology. So as we went on, we came across a lot of limitations with that particular technology. Like we cannot expand it, we cannot scale it, and you have to use a lot of energy to get the water from those missions. And we cannot take, when all these limitations are available, we cannot take the water to the mass population, or rural populations where energy is not very easily available. And when you cannot scale it, you cannot take it to the community. People only who can afford to pay for the energy or pay for the missions, they only can have it. So we started exploring other technologies where we can overcome these technical difficulties. So since 2012, we started researching on other technologies where we can scalable from 10,000 liters to 10 lakh liters of water using only very limited amount of energy. For instance, this new technology enable us to take water from the ground and process it like RO or anything. It is cheaper than tapping the water using our technology. So this is the um, what is it?
Breakthrough.
This is the new breakthrough. Like we are trying to commercialize among different stakeholders, including government and people. Especially, we are focusing on the arsenic population. In India, almost 8 states are highly affected arsenic population. Almost 60,000 habitations have no water resource. They cannot use the groundwater or the surface water. And when you put any arsenic filter, then the disposal of arsenic is a huge threat. You cannot dispose it again, put it back in the ground, or you cannot throw it in the sea. There is no way of disposing the filtered arsenic. So we have to introduce them, give them a new source to tap the water. And that's what our technology, in our technology, we are trying to do. And
Yeah. The two ways in which we can think, or three ways in which we can think in terms of this technology and this innovation of ours paying off in terms of social change. First and foremost, we are getting people to access water at a very low price. It costs us something like 19 paise to produce 1 liter of water from air, which is cheaper than the cost of pumping the water from the ground, putting it through an RO machine, and extracting the water from therein. It's much cheaper than that. The second area in which we are promoting social change is by providing a water which cuts across all kinds of social differences. For example, this water is accessible from the atmosphere to whether you are an Ambani or a very poor man in a slum, because the water quality remains the same and the water quantity per unit of energy still remains the same. So this is across economic differences. Secondly, we also have a way in which we can ensure excellent health and high productivity for the population. As Jennifer just now told you, 60,000 habitations are arsenic affected in this country, which means they are on the verge of or on the way to severe carcinogenic disorders of the body. In fact, the disorder is so heavy in eastern part of the country that Bangladesh hosts a major arsenic research center by the WHO.
we call it
Now,
equitable water.
right, and then you get equitable water all the way, whether you're rich or poor, whether you are in the rural areas or in the urban areas. The same atmosphere which is spread all around us gives us as much water as we want at a very low price. So the last point on the equitability issue is which caste you are, because in rural India it's a major issue whether you belong to caste A or caste B, and whether you can use the water resources belonging to one and hand it over to the other. Now, this issue is simply bypassed in our technology because everybody taps into the same air, and the village can have a water plant which everybody can access and which can produce just 5,000 liters a day or 5 lakh liters a day, it's accessible to everybody. So these are the three ways in which this technology and this innovation provides the ground for making social transformation. It's well in line with our old argument that the first major social transformation, civilizational transformation, began in pastoral communities when water management arose. Long after all that happened, today we find that we have access to atmospheric moisture, which provides a similar access to social and civilizational transformation. We are open to questions now.
Thank you so much, Dr. S.S. Shivkumar and Madam Pandian, for that extremely eye-opening presentation. And on such a vital issue, which affects everyone, like you said, across socioeconomic strata, and in a country where water scarcity is one of the core issues, both today and going to be so increasingly in the future. So thank you so much for that insightful talk. I now invite the audience. So I am joining online. So I will be very happy if the audience members can either raise their hands or type in their queries in the chat box. And I request the organizers, for those people who have joined us in physical mode, to kindly take questions if there are any from the venue itself. So please, the floor is open. Meanwhile, sir, I would have a question. So
Certainly. Go ahead.
Yes, sir. So we just want to know whether, what kind of funding has the government, how has the government not lapped up this technology, given the water crisis that is facing our country, and given the huge advantages in terms of equity, in terms of costing, in terms of access and availability, that you have made a breakthrough with. This is transformative. So why has the government not really taken this up in a much bigger way? And is there something in the pipeline for people? So
Yeah, very briefly put, you see, all big change in society is at heart of it all a political process. And the governments, in a sense, are the catalytic agents of such political processes. At the point in time at which I'm talking to you about it, the government has responded to us as a startup, and a certain grant has been made so that we could make a pilot out of it and a public demonstration. But as things stand, the government still is to open itself wide open for this project.
I would like to add few points. On a policy level, the Ministry of Drinking Water, and they have certain parameters, like to provide drinking water or anything, it has to be within this price range. Any fellow sitting in a rural, they fix the limit. Everybody have to get 40 liters of water. That is their target. But that 40 liters is to bathe or to drink. There is no classification. Only recently they made this classification. Now they are saying each individual have to get 4 liter. They made the differentiation between drinking water and water to general use. So that one. Second, as I mentioned, this limitations, you cannot scale it and it is expensive. So government were really not keen to look at this. Only in 2016 they formed a committee called the Mashelkar Committee to look at very innovative different technologies, where we presented our technology. Only after that they started including this option in a policy level. So this year the Ministry of Housing and Urban Affairs, they called for India Water Pitch. So we participated in it, and we got the grant to set up a pilot project. So they, it is in the process. Once they see this, they want to scale it up to the 500 smart cities to use the same technology for offices and institutions like hospitals, schools, to for the drinking water needs. And I would like to add, just last week we signed a MOU with the Directorate of Public Libraries, which manages around 4,500 libraries in the state. So we together took the climate pledge that when we set up this 2,000-liter system in their office, every day they will be saving 6,000 liters of groundwater, because each liter from the atmosphere saves 3 liters of groundwater. So every day it is 6,000, and for a year it will be like 21 lakhs liters of groundwater will be saved. And this technology is net zero enabled. So they will save around 5 lakhs something CO2. So this technology easily combines with the recent, climate action
Right.
Scenario.
Thank you, ma'am. So we have a question in the chat box. Let me read it out for our speakers. This question is by Aishwarya Goyal. She wants to ask, what technology and methods do you actually use to tap the water? Looking at the huge size of water bodies and extensive infrastructure required to cover all of it, is it something that we can also install at our domestic or personal level usage?
I'd like to answer this. You can touch it.
The technology and methods.
Okay. Okay. Well, Aishwarya, thanks for this question. It's a very important question. In answer, I can say this, that the technologies which we have at our disposal and which are our patented technologies, we do make machines which you can use in your house, extract water from the atmosphere for your domestic use. That is there. But we are now on technologies which are mass level technologies, as I said, where you can, in a village, produce 10,000, 20,000, 50,000 liters of water as they need from the atmosphere. All this, the whole range of, a whole spectrum of technologies are available today for this.
Aishwarya, I would like to add something with what Doctor have said. So when it comes to technology, I can easily, name it as vapor compression or it is heat exchange or something. But I would like to, give something so that you can imagine or relate. For example, if you take the chilled water bottle from the fridge and you take it out and put it under the fan or something, when you let it expose to the atmosphere, you can see the droplets outside of it, right? So, uh, are you there, Aishwarya?
You just speak.
No, I want to make sure, is she listening or
Yes, ma'am. Yes, ma'am.
is
She's
she?
listening. She's also
Yeah.
responding in the chat.
Okay.
Yes.
So that is the same process. Like you have to cool the surface, and which method or technology you use, that differentiate the cost which involves to do the process. In other words, the energy amount of energy, so which directly relates to the cost. So we have technology which can be used at the household level, but again, that energy consumption will be more. So that's why we stop selling those machines to the people, individual people or institutions. We use this technology where we can produce large amount of water. So we are selling this water in in a way which people already used. Everybody now buying this 20-liter cans, either Bisleri or Aquafina or any brand like Kinley daily. So we are going to give our water in that packaged 20-liter bubble top cans or in the bottle, which people already conditioned to use it. So in that way, people can save lot of capex and installing these machines, AMC, and to run the machines they have to pay for the energy cost. It's all removed from the customer point of view, yeah, portfolio.
Right. Thank you, ma'am. She also sends her thanks for this.
Yes.
So we have another question by Dr. Tejendra Kumar. He's a faculty at Maitrey College. He asks, sir, your intent to provide water at such a low cost, but as you mentioned earlier, government is not providing the requisite support. How will you be able to get your funds so that the technology you are using is well taken care of? Because if you're not making much profit, How are you ensuring the good condition of technology? I beg your pardon. He is asking this question on behalf of a student from Sociology Honors. She's called Pari.
Excellent. So thank you very much, Dr. Tejendra, and the student whose name I don't know.
Pari.
Pari.
Pari?
Pari.
Pari, okay. Pari. Okay, thank you very much for this question. There was a bit of a confusion about the name. I called it Pari because same Pari is the name of my son. He's a boy.
Oh.
So anyway, thank you, Pari, and thank you, Dr. Tejendra. Uh, the question is of government support. Uh, you see, you can't do any of these things without you make a business out of them. The government can support with financial help, subsidized interest rates, and various other aspects of the market where they can intervene and help on your behalf. But eventually, doing this as a business and making by the water and make a profit out of it, you're absolutely right. It's our job and our business. So if we got a good business plan there, and we are moving along nicely in that. All I can say at this point in time is that we are the cheapest source of the purest water in the market. I think we are extremely confident that we are going to go right ahead and move along in the market.
So in this forum, we are talking more about technology because we haven't touched upon the product. The product we are going intend to introduce in the market is natural premium water, like the Himalayan water or Vedika water, which you get it in the airport. So we come under this premium segment. There are only two segments the government of India certify for packaged water. One is normal packaged RO water, that is the 20 rupees bottles which you buy Bisleri or coffee now. Another one, this premium segment. This is 100 rupees bottle, you get it in the airport. Because the premium segment, you have to, pack the water at the source. So we come under this category because in our technology, once the machine produce the water, immediately it will be packed. So we come under that category. And first time, people, when they subscribe Bisleri 20-liter can to their houses, nobody selling at this point in the market, no premium water sellers, they are not giving water in the 20-liter bubble top cans. We are planning to introduce that premium segment for this B2C customers. For the officers, they are buying every day hundreds of 20-liter cans from Bisleri. So now we are going to give them the premium segment water at the same price. And we are going to attach this carbon credits because it is a net zero enabled technology. So when we process to get this product, we can earn carbon credit, which we are happily, assigning to the customer as a kind of incentive. So that's how we know the business is going to make money or the market share that we would like to capture.
Thank you, ma'am. So, I think we are all very certain that this venture is going to grow from strength to strength because of all the advantages that it has, and also it is such an important step towards sustainable change, which is also equitable, which is having all the merits that one can hope for this issue. So let me see if there are any more questions. Okay. So again, sir, this is not a question. I hope that we can share if you will be, if somebody has, later questions or follow-up questions, or someone wants to read up on your work, is it okay if we provide your email IDs to interested students or faculty members?
Certainly, you may. Certainly.
Right.
I just type it, the email, so you can share it.
Yes, ma'am. Yes, ma'am. If you could do that. Also, as an aside, so this has obviously been developed for the Indian context because this atmospheric moisture extraction technology that you have pioneered. What kind of conditions is it? Is it amenable to all kinds of climates? So across geographical borders, or is it a certain kind of climate that takes it to make this more feasible as a technology to use? For example, if we have certain areas which are very dry, will this technology still work? Please pardon me if it's a very
Excellent
basic question. So,
No. Excellent question. It's a very basic question which faces us all the time. Uh, by and large, this technology is good for tropical climates, and it works best in a country like India within a 100-kilometer distance of the oceans. That is basically 100 kilometers from the Bay of Bengal on one side, 100 kilometers from the Arabian Sea on the other side. So, by and large, it's not very good when the atmosphere is very dry and very hot, such as like central India, Madhya Pradesh, Bhopal, such are the places. However, the good thing about India is that we also have monsoon rains. So when the monsoon rains are on in the country, when the air is very humid, so there's a good 5 month period when you can extract as much water as you can from the atmosphere. And whether you're in Bombay or whether in Bhopal or in Chennai, during the monsoon season, there is water available in plenty wherever you are.
right, sir. Thank you so much.
Thank you.
Devangna, one student has a question. Can we take one more question?
Yes, please.
Sure.
Yeah.
Good morning, everyone. I'm Hadia Arif from Sociology Honors first year. Uh, as someone as someone pursuing sociology and being quite ambitious about bringing some or any sort of societal change, so your approach has really motivated me. And I would be obliged, and also my peers, if you enlighten us about how you were motivated to start this project.
Well, you see, we are all Indians, and we share a huge cultural and traditional heritage. So you wouldn't mind it if I tell you how it started? It started in the year 2003 when Sri Satya Sai Baba told me to go and solve the water problem of India. And I didn't know what I was meant to do. A week later, he came and asked me if I was confused. I said, yes, of course. And then he told me, the water in the ground is all dead and gone. It won't last another 10 years. But in the air, there's all the water you want, go tap it. So that's what put my mind towards atmosphere and that process. Eventually, I went back to my university in Madras, where I taught in those days, and told my vice chancellor that I want to go and do this, so kindly give me voluntary retirement. He was happy to give me a voluntary retirement, and I went out, started a factory, and in about 6 months' time, we had the first prototype up and running, and in about a year's time, we were in the market. What I'm trying to say is there are all kinds of things which going to push you into a decision. In my case, it was Satya Sai Baba. It could be anything else in anybody else's case.
And I've been student of Dr. Shivakumar for last 10 years. So when I studied his earlier work from 80s, his papers and his experiences, he wrote about all sorts of things when he was a consultant to World Bank and Government of India. He used to tell us when he teaches things, or he used to tell us from 1980s onwards, I knew there was no water in the ground to manage. So there is no concept of managing water. But it comes to his mind later when he got his inspiration from his guru or his
Sai Baba.
yeah.
Yes. So, well, I do not know how this way of starting research and an economic activity would appeal to most people, but I think being Indians, most of you would understand that it's very reasonable that a guru tells you to go and start a business and then you go and start a business. It happens. That's what happened to us.
Thank you so much, sir. I think it's a extremely engaging and a very inspiring story. And you're right, it just takes one spark to ignite that entire idea into something tangible. And I think inspiration is all around us, provided we are amenable to looking for it. So thank you so much. Yes, I will also share this email ID and the PDF that you have just shared with our students and our peers so
Yes.
that we can you know, come to you later via email or otherwise.
Yeah.
So I think
You know,
now,
Jennifer has just sent you
Yeah, that's what she said.
a presentation which you might like to share with students and others in around you there.
certainly, sir. Yes,
Thank you.
thank you, sir. Um, right.
So we have one last question.
Okay.
Sir, I have a question to ask. So, whenever we talk about water scarcity in India,
Uh,
the one question that comes to my mind is in India, we have places which are affected by flood, while in the same country has places which faces water shortage. I think, sir, it would be better if we have an equal availability of water throughout the country.
yes.
We have projects like river diversification, water linkage, but is there any such technology or system that can that we can adopt or the common people can adopt so that we have equal availability of water throughout the country?
Yes. Uh, but may I know Devangana Jha? Am I right about your name?
Yes, sir.
Okay. May I know what the question here is?
Right, sir. I'll ask Apurva to again do it. Yes, sir. I'll just ask
Yes.
Apurva to repeat the question. Apurva, could you rephrase the question since this has been asked in the physical mode? So I
No,
will
no, it
ask
doesn't
you.
matter. If you can speak it out again, it's fine.
Right. Apurva, can you ask the student to repeat her question, please?
Sir,
Yeah.
sir, is there any technology that can be adopted by the common people so that there is equal availability of water throughout the country?
Oh, yes. I mean, that's exactly what we've been trying to do for the last 20 years. And I'm happy to tell you that as of the last couple of years, we have hit a gold mine in the sense that we have found a technology which is what you may call dirt cheap, and at the same time, it can operate in all parts of the country. In some parts, better, in some parts, not so better, but it can operate everywhere. Did I get across?
Yes, sir.
Did I,
So
Yes, sir.
did I get across? Hello?
I hope it is clear to the query is clear. It's now answered. Yes?
Yes, sir.
All right. Yes, sir. She has
Thank
understood the
you.
point. Thank you. So now there are no more questions. So we conclude, we comes to the end of this very invigorating session. Let me again extend my warmest thanks and gratitude to both our speakers for sparing their valuable time and for apprising us of this very important and very interesting new breakthrough towards water management and towards solving the water scarcity crisis that is looming in our country. I thank all the students, all the teachers, all friends who have graced this occasion, both by joining us online and as well as in the physical mode on the college premises of Maitrey College. I hope that you all remain with us for our final valedictory session that will begin shortly. And you will be able to listen to more experts and erudite scholars talking about various issues of resonance with society. So once again, thank you so much and have a good day. Thank you.
Thank you.
Thank you and namaskar.
Namaskar, sir. Thank you.
Hello, good morning, everybody.
Hello, ma'am. Ma'am, you are audible to us. Am I audible, ma'am? Ma'am, you are audible. Am I audible to you? नहीं हो रहा.
Stu- Suman Gobil, ma'am, can you hear me? Hello, ma'am. Am I audible? Hello. Am I audible?
This is Dr. Majumdar. Can you hear me?
Can't hear you.
Hello?
You can hear me?
A very
Yeah.
good morning to all our invited dignitaries, learned members of the teaching faculty, participants, and dear students. It's an honor and privilege to welcome you all to third and last day of the annual international conference, Equinox 2023, on research development and social transformation under the aegis of Vantage Journal of Thematic Analysis. I have a pleasure to introduce Dr. Suman Govil ma'am, who has very graciously agreed to chair the valedictory session of Equinox 2023. Dr. Suman Govil obtained her master's with first position from Delhi University. She did MPhil and PhD from Delhi University itself. She was involved in key policy decisions and important initiatives of the Department of Biotechnology for 36 years in the area of human resource development and preparation of strategy. Known to be proactive, social person, she believes that quality manpower holds the key to success in every area. This has motivated her to work on education and training projects that aim to produce world-class manpower in biotechnology in India. She has initiated postgraduate and postdoctoral teaching programs in biotechnology in more than 70 universities in the country. Her pet project, the Star College Scheme, has enabled the integration of research into undergraduate science education. Through her blog, Opportunities and Concerns in Biotechnology, she communicates information about latest policies, schemes, and shares exciting science news. Dr. Suman Govil, ma'am, I hope you can hear me. Ma'am, please take over the session and introduce our honorable chief guest, Dr. Subir S. Majumdarji. To you, ma'am.
So organizers, can you hear me? Hello?
Yes, sir.
You can
Yes,
hear
sir, we
me?
can
Huh?
hear you.
Okay, okay.
Yes, sir, perfectly.
Fine, fine, fine.
Suman, can you hear us?
So I think Suman madam has some issue.
Good morning, everyone. I hope you can hear me. It's a great
Yes,
pleasure
ma'am.
for me to be here amongst you and to be able to introduce one of our own scientists, Dr. Majumdar. He's a highly renowned and ornamental, decorated scientist. He's a member of all academies of science. He's done path-breaking work in the area of male fertility and has published in reputed journals like Nature Medicine. So I would not like to come in between you and him. So without further ado, I request all of you to join me in giving a huge round of applause to welcome Dr. Majumdar. Dr. Majumdar, all yours. Thank you.
Thank you, Suman madam. And we have long association of DBT. And also thanks to organizers of Equinox. And I am happy to be amongst you. And I will try to share my screen. Is the screen shared now?
Yes, sir. Yes, sir.
Okay. So good morning to everyone. And I wish good luck for all of you, especially students who are sitting and listening, and hope you do great in future. And I have made the slide so that it is student friendly. So I will be talking about generating human therapeutics in milk to reduce the cost and increase the affordability by masses. And these are my students, Abhishek, Gautam, Sovik, Nirmalya. And this work consists of majorly the work from National Institute of Animal Biotechnology, and also from now I am in Gujarat Biotechnology University at Gandhinagar. So different proteins are required to be supplemented as therapeutic agents to certain patients who are unable to produce them naturally, or when excess is needed for a treatment. For example, if there is a burn injury, body produces IgG, but you need to give excess IgG. When there is a Guillain-Barré syndrome, you need excess IgG to be given. So it's not necessary that it should be producing less. Sometimes it is given as additional therapeutics. Such proteins are produced exclusively in bacterial or mammalian expression systems in vitro, in industries, with big fermenters and all. To tackle high production cost, various attempts have been made to create transgenic animal bioreactor for expressing various therapeutic proteins in the milk. So other countries are doing this kind of work, trying to help out by making such therapeutic proteins in milk. But it's very difficult. Let us see first what is the problem. So if you go to buy therapeutic proteins, you will find several therapeutic proteins. And since medical science has witnessed a great enhancement in their knowledge about protein-based therapeutics, so there are a lot of therapeutics proteins which have come in the market. So let us say Guillain-Barré syndrome. So the outer layer of myelin layer gets shredded off because of the infection or post-infection, and then you need to whack in a lot of IgG to prevent bad antibodies to work on them, and that's very costly, 12,000 rupees a vial. For several kind of inflammatory disease and arthritis, you need bone morphogenic protein that is also extremely costly. So people usually don't use except for very rich people. Now the cost goes in the range of, you can see I have given that for insulin, blood coagulation factors, interferons, growth hormone, bone morphogenic protein. So cost really ranges very high. You can see sometimes that each vial is 1800 to 2700, and a treatment for 6 months cost around 3 to 5 lakhs, which poor people can never think of using. That's next to impossible for them to really even think of using them. So what is the alternative? So alternative to us best is that this huge cost, if we can, instead of industry, if we can make an animal bioreactor, that would be best. So I will try to explain you what I'm trying to tell you. You need a huge land, you need a factory, and then you need huge infrastructure, electricity, labor, working in various shifts, licenses, all these kinds of things you need. And for that, the huge cost is built. But if we can do by making such transgenic mediated therapeutic proteins generation in the milk of an animal, you will find that this animal itself becomes a standing industry. It is your light, electricity, land, license, everything. You feed, everything is covered. 3 shifts of the workers, everything is covered. And also, as compared to the bacterial or other expression system, mammalian system is far better, and glycosylation, etc., goes on very good. So at any cost, if we can generate such animals, it will be very, very cheap as far as the products are concerned. So if now, since there are students, let us talk about transgenesis, how to make a big transgenic animal. Transgenesis involves insertion of genes in the genome of plant or animal to obtain desired trait. For human health, you can generate a genetically modified organism, and that is used for basic research at today's days, mostly for humanized diseases like diabetes or several kinds of diseases, which I will not name here. With the advent of the high throughput tools of the next generation sequencing, we are learning about many new genes based on their sequences. However, their functions need to be evaluated in whole animal model. You need the full animal. With modern tools of bioinformatics, researchers are really able to assign functions to several identified genes. And because you assign functions to proteins made by these genes, you can do various experiments. And this has further proven role of certain genes or proteins made by them in a particular disease or alleviating sufferings from a particular disease. So basically, you can take a human gene, insert in a mice or rat, and generate a transgenic animal. And this is called humanized disease mouse model. That's a standard transgenesis. And for students, you must learn that whatever we talk about a gene is nothing but a machine reads a sequence and starting codon and termination codon. It is called as a gene. Machine doesn't know what is its function. So you have to insert this gene into a system, whole system, and know the function of it. For making the life very easy, let me say that the promoter of a gene is an engine where the machine starts reading and says, okay, gene has started. Then there is actual gene which is converted into RNA or transcription factor or whatever portion. But this is a promoter which drives this gene. So promoter is tissue specific. So these genes may be present in all parts of the body in our nucleus, but only in brain, for example, if there is a GFBP promoter, so glial fibrillary acid binding protein promoter, the promoter will work only in the brain. And therefore, that protein is produced in the brain. Now, if I tag a different gene behind this promoter, that gene will be expressed in the brain because promoter works only in the brain. So remember, promoter drives a gene tagged behind it in a particular tissue. That's very, very important for you. So mostly transgenesis is done when fertilization occurs and cell divides heavily. At that time, a gene is inserted. So how gene is inserted? So this is a fertilized egg. You can see male and female nucleus, and we insert foreign gene from right in a small glass tube, and only around 4 picoliters we inject into the male pronucleus. Now, when the cell divides, we pray to God that this inserted gene, see, this is swelling. You can see it very easily. That this inserted gene should integrate into the coming mice, and that mice becomes transgenic. It is not that it always happens. Out of 100, maybe 4 or 5 such wonderful genetic integration occurs. So you see, you can catch another, and you can see a moving sperm on the lower side. And again, he can inject the gene of interest. So this is the way basically transgenesis is done. Other way of transgenesis is cloning. That means if you can remove the nucleus of this is a buffalo embryo. We worked with buffalo embryo also in past. If you remove the nucleus from this egg, then egg will become nucleusless. Now, if you insert a transgenic nucleus, then animal product will be transgenic. So that also can be done. So I will show you how this works. So this is typically by which Dolly the clone was made. So this is polar body. I'm not interested. This is hexadyne staining. So you can see the from inner side how the nuclear material of the egg is removed. And once it is removed, this becomes a enucleated egg. Now, these are the cells which are transgenic cells. These are cultured and gene is inserted in them. Their nucleus can be inserted into this egg, which is the enucleated egg. And then egg can be activated electrically or by chemical strontium chloride, etc. And then this egg will start dividing, and then you will get a transgenic embryo. So let us see, these are the cultured cells of the buffalo oviductal origin, and these are transfected. So there are several cells, but only few are transfected with green process protein. So if you remove this nucleus and insert into enucleated buffalo, ideally buffalo embryo should turn green. Buffalo embryo is otherwise not green. So see, when we have done, we found that out of 2, you can see the inner cell mass and trophoblastic cells. So this transgenesis is possible by cloning also. This we published. So either you do the insertion of the way I told, or you hold for 1 second. I want to lecture actually. So this is the only way that transgenesis can be done, either by male chronicle injection or by cloning. You need very costly equipments, and you need basically several females to be superovulated to make 2 or 3 transgenic mice. So this is a cumbersome procedure. So we started thinking, can we have some alternative procedure to this the same thing? So what we found that basically it is the cell division during crossing over, that when these kind of changes are occurring, we can insert from exogenously some genes, and this gene can by chance get integrated, because that kind of things are happening at the time of heightened cell division. So we started thinking that this procedure is like if we invite some famous dancer for dancing, let us say we call Madam Hema Malini for dancing. It's an organized structure function. There, if some gentleman goes and jumps and says that I am very excited, I also want to dance with her, it's not going to happen. Police is going to come, hold him and take him away, because it's an organized thing. So my skin, for example, surface is an organized place. If I put genes there, nothing is going to get integrated. It's not possible. But let us see another scenario, that in a disco, there are people dancing and enjoying their life, and band and sound is coming. There, if this gentleman goes and says, I want to dance, he will be easily integrated and allowed to dance with them. Why? Because there is a haywire system going on. There's a huge cell division going on there. Nobody cares who gets an integrated here. So we thought of this, that during embryogenesis, this is what the investigators before us encashed. At the time of embryogenesis, they inserted the gene. Nobody took care of who is coming and dancing with them, and they integrated. Now we did think that in case of spermatogenesis, a human being produces around 200 million sperm in one day. So much of mitosis and meiosis is going on. Why not to exploit that rather than egg? Because that takes so much of time. There is no guarantee. Then you have to kill around 100 females to make 2-3 transgenic lines. Why to do that? So we went for that, and we took one male. And if this is successful, then we do not have to kill anyone. Just if the stem cells of the testes are integrated with this gene, you simply put this male for mating after 30-40 days. It will keep on churn out, making some transgenic sperm, and normal mating should result into production of transgenic animals. So we did use the testes. You can see the seminiferous tubules of the testes, and our goal was to hit the stem cells of the germinal origin. And we did an electroporation in the testes. Tryptophan blue is given to ensure that there is no leakage. And we gave an electric current along with the DNA inserted. When you give electric current, the stem cells open a mouth, and this solution consisting of our DNA particles will get inside. And automatically, because cell is dividing, there are chances of integration. This is just to show that electroporated testis is green, non-electroporated testis is not green. That means when we inserted gene, the genes were expressed, that of GFP. And then we put for mating. And then we did series of experiments to prove that this works, and published this paper in Nature Methods. Then instead of surgery, we found many people have problems. So we now standardized a procedure where both the testes can be injected. No need to do surgery. From external electroporation, you can generate transgenic mice. And we published around 14-15 papers based on this technique. And this new technique is also good, because if we can take human keratin 14 promoter and tag EGFP below it, as I said, only keratin, that means the skin layer, this promoter will work. And since I have tagged EGFP, you can find this is the whole section of the skin of a mice, and you can find no other place there is a green fluorescence except for the keratinous layer. And see wonderful presentation showing external electroporation also works. So this new method of electroporation where germ cells are integrated into, the gene is integrated into male germ cells is very, very beneficial. And now you can think of making a large transgenic animal. Because using embryos, you need to have 100 buffalo standing for superovulation and another 50 for embryo transfer. That's just not possible. Nobody is going to give us money. So basically, this kind of testicular electroporation can be used in large animals to generate a bioreactor so that therapeutic proteins can be expressed in the udder gland. Now, when I talk about udder gland, that means I now need a promoter which is of udder gland origin, so that anything I tag in the udder gland origin promoter, if I tag human gene, the milk in the udder gland will drive that gene and make human therapeutic protein in the milk. That's my target. So then we can use this buffalo or cow or goat as a bioreactor. So for that, first thing we did is we isolated the promoter of the buffalo beta casein. Why? Because goat beta lactalbumin, goat and cow beta lactalbumin have been isolated by foreigners, Western countries, and they are trying to make therapeutic proteins using that. So we thought buffalo is indigenous to Southeast Asia and nobody isolated. So we isolated buffalo beta casein promoter to avoid the patent related thing. And successfully we tagged EGFP below it. And then our first job was to see that this IRS2 EGFP, whether this EGFP expressed in the cells or not. So we took this construct, which is isolated buffalo beta casein tagged to EGFP, and cultured mammary epithelial cells and also Echekithre cells. So only mammary epithelial cells where this promoter can work, because promoter is udder gland specific. It expressed EGFP, not in the Echeke cells, which are the kidney cells, right? So now if we need to generate large animals, what we have to do, first we generate a mice. So we used external testicular electroporation procedure to generate a mice. Now what we have to see, is it that if it is a EGFP tagged gene, that my udder gland is green, no other portion is green, heart, lungs, kidney or anything. So also do Western blot to show that the protein of green fluorescence protein is not expressed in other parts of the body. So we made this transgenic mine, and in lactating udder gland, we found that if I cut open that mice, and if this is the udder gland, you can see udder gland is the udder duct. Other portions which are inner side of the skin, they are black. You can see this is black. But only udder gland with this duct, that is turned green. This is one udder gland, that is another udder gland. So that means it is very specifically expressing the gene I tagged, that is green fluorescence protein, only in the udder gland. It is not going to any other part of the body. Now we have to kill the animal. So we did so and took out the spleen, liver, kidney, heart, mammary gland and brain, and found only mammary gland is expressing EGFP, no other portion. That means there is no leaky expression when we tag with the buffalo beta casein protein. Understand here, students, several promoters work across the species. So buffalo beta casein promoter works across the species and it does the same function in mice also. Now the problem is, if I generate such a large transgenic animal, will it be accepted? We know that there is a genetically modified organisms. People talk about risk, future hazards and all these things. So there is a major issue. While we are keeping on working on this line, tomorrow somebody will say you cannot generate a genetically modified organism. That means germ cell should not have the foreign gene. So answer is directly inject the breast gland. Don't make a testicular or egg related or embryo related work. Just go directly into udder gland, transfect it. If somebody steals my animal, it cannot reproduce because germline doesn't have the gene. So directly inject the breast gland with something to transfect the gene. And then what we used virosome. So this is a virus. This is the nuclear material of the virus. Our job is to remove the nuclear material of the virus and to insert the DNA of our interest, that is buffalo beta casein promoter tagged to human gamma interferon, for example. So we need a new DNA material inside. So that is done. There is a procedure I have not shown because of lack of time. So membranes are cut, DNA is, RNA is thrown, whatever virus. And then when you remove the what you call the soap or the detergent which has broken open the membrane, as you dilute it, remove it, the membrane try to come together. When membranes try to come together, you insert a lot of foreign DNA there, so they will be trapped. So now you have a virusome which will dock on the cells of mammary gland and will transfect it, but the DNA is what we provided. So we can successfully do that. So we did it, and we injected in the udder gland directly. You can see it very nicely. It can be injected. It's not that difficult as you may envisage. See, and then the beauty is at what stage of development of the udder gland you do it. Every species it is different. So we have standardized them, and what we found that if you do that, basically you inject this and the solution goes inside, and if it is a dividing udder gland, again like testis, it will integrate the gene, and when it integrates the gene, it will express the protein. So that was our job. We did it successfully. Again, we killed the animal and found heart, liver, kidney, spleen, brain, abdominal doesn't give anything, but breast gland definitely expresses EGFP. So that was a very successful thing. And now we want to not use EGFP, but human gamma interferon for that work. So we used human gamma interferon as a protein behind the buffalo beta-casein protein. And these are all controls. See, nothing is expressed, but with our virus of 100 microgram and 75 microgram concentration, when we transfected, we found there is a production of human gamma interferon into the milk. It was very good. And then we have seen this into the breast gland. We macerated the breast gland and checked, and we found as compared to wild type, there is a lot of human gamma interferon production. And then we have taken out the milk. This is the way to take out the milk, either with a buffer or directly you can hang it. And this milk which is collected, we did ELISA, which is used for human gamma interferon in the market. So we procured that and we checked in mice milk, can you get human gamma interferon? Luckily, what we found that in wild type animals, not transfected, nothing was detected. But in the milk, also we found that there is a gamma interferon production. It's a very good thing. And next, we should work with goat, buffalo, cow to obtain large quantities of therapeutic proteins. So remember, there is a lot of bovine FSH and bovine LH, which is supposed to be used in overcoming infertility in cows and buffalo, but it's so costly, nobody can use it. It is used for human IVF. So we have now produced this bovine FSH and LH for our farmers into goat milk successfully. And what we found that not only that it is produced, but it is also working functionally. So stem cell factor and FSH receptor, when cultured Sertoli cell are given the bovine FSH from the milk gland of this rabbit, they are showing actions as bovine FSH does, same way recombinant bovine FSH is in our hand doing. That is stem cell factor enhancement as compared to the controlled Sertoli cell cultures. And so with the FSH receptor, that is the typical action of the FSH. So not only that now we are producing proteins, but they are biologically active also. So that's a very good thing for us. And 16,000 milli-IU per ml of rabbit milk, we got this. It's a very encouraging thing. And now the virosome cost is very high. So I'll go to another. Now from GMO, we came to non-GMO animal. Now virosome making is very costly. 400-500 eggs at a time. Every batch is not successful. Several times you fail and the money is wasted. Poor farmers can't afford that kind of cost. So now what we have done, that in the udder gland, when you are trying to transform, can we think something else other than virosome, so that a costly virosome can be replaced? So what we have done, now we have generated a hexonylated branched polyethynylamine. So branched polyethynylamine is used for transfection, and it does some level of transfection, but you, when treat it with hexanoic anhydride and make a hexanylated branched polyethylamine, its intensity is heavily increased. And this we have filed a patent for it. And now we have shown that as compared to BPEI, there are more amino terminals used for the binding of the hexanoic anhydride, and FA6-BPI is better than BPEI. And what we find that when we do the transfection, this is a untransfected control. There is no EGFP expression if the gene is EGFP. This is lipofactamine. And below the lipofactamine, you can see the BPEI or alone. And then this is FA6-BPI. So you see, if you see the expression, even the cytoplasm is full along with nucleus and everything. So expression is far better as compared to lipofactamine or plain BPEI. Now, when we go to, this is the same thing now given as a n is equal to 3 experiments, and everyone where it is shown that FA6-BPI is far better than even lipofactamine, which is available in the market. And this is in-house made, so much cheaper. Now, when you go and transfect various cells with the CMV promoter and EGFP, they should transfect all cells because CMV is expressed everywhere. And we found that BPI6 is always far, far better in terms of the expression of the tag gene. So BPI6 is very, very cheap. In less than 2-3 rupees, you can transfect the gland because it's just a chemical, simple chemical diluted solution. So really it has come down, cost has come down. And we are very, very happy that not only that we are transfecting, but viability is also very good. So you can see in 293T cells or MCF7 or even goat mammary epithelial cells, the viability is as good as that with the lipofactamine. And we are very, very happy that there is no toxicity as it's a chemical that was the issue. This is nothing but the same figure compiled together showing that there is no cytotoxicity in FA6 BPI usage as compared to any other method. And I'll mention that this was assayed by using MTTSA, and we could successfully produce, you can see human gamma interferon through the cultured cells when CMV was the promoter. So all of them expressed. So this is working good. Here, the purpose was not to use buffalo beta casein promoter, but here the purpose was to use the agent to ensure that the mammary epithelial cells are transfected well. So then now we have transfected goat. I have not given that photograph of goat because several times people get excited or they comment, why are you injecting this in udder gland or all? But that transfection in the goat we have done, and these are the milk samples. These are 24, 40, 48 hours. Up to 72 hours it is secreting, but then it is falling down. So what we need to do, work on this to see that if secrets for longer duration. Even this is good enough, but we will try to see that now we have gone to higher animal. With HABPI6, F6, we have injected udder gland of goat, and we have successfully generated human gamma interferon in the milk. This is a wonderful observation for all of us. If this happens, really, poor people can be given. So take-home message from all of this is from human genes, you can tag out or bring out a gene which is very, very important for a therapeutic protein. You can insert that gene into a transgenic bioreactor cow. One of the ways is doing that, so that a costly protein of around 14,000 rupees or so can be really brought down to rupees 200 a vial, because whole industry, my cow is the industry standing. As I said, light, electricity, manpower, labor, license, whatever you call, right? So now this is very, very important for us that we go. But if we do transgenesis in large animal, we have to remember in mice, if you put for mating, you produce 12 babies. Out of that, 2 or 3 are transgenic, you are fine. In case of buffalo, you have to produce, you produce only one calf after 10.5 months. And if that is not transgenic, you're gone. So what you need to do there, when you transfect the gene into the testis, what you need to do, ensure that you tag a protein so that your sperm is green. Your sperm is green, and your sperm, which is green, it's a bicystonic construct. One construct is to make its surface green, and continued construct is the human therapeutic protein. So only green sperms can then be isolated using, let us say, tag your gene in such a manner. There are 2 promoters. One promoter to drive on the surface of transgenic sperm greenness, another continued bound stitched promoter, which is a promoter of buffalo beta casein tagged to say human gamma interferon. So every green sperm have the gene of my human interest. So I can separate them by fax. You can see it. And when the green sperm come in one of the vials, you can use it for various kind of functions, or you can do the magnetic separations also. And then this green sperms can be specifically used for several kind of things, either for intrauterine insemination or for IVF and embryo transfer in cow or goat, or for intracytoplasmic sperm injection to generate a transgenic cow. If you want to do transgenesis, you have to tag the sperm, because you can't say let God do the job that my transgenic sperm goes and fertilizes. You can't. You have to isolate transgenic sperm, because after 10.5 months you'll come to know whether cow or goat or a buffalo is transgenic. But God forbidden, if you can avoid this procedure and go for non-GMO, that is, take a cow or buffalo or a goat, directly inject into the udder gland the gene of your interest, so that nobody is going to fight with me. why you made GMO animal. I am not making GMO animal. My udder gland is transfected. Anybody can steal it. They cannot reproduce because germline is not getting anything. And such, if we can drive several human therapeutic proteins, that will be wonderful for this country. So that's what I wanted to share with you today. And thanks to the organizers of Equinox. And I would say all the students, keep your dreams really very high, very divergent. Think innovatively. And I'm very sure out of all your work, one balloon will reach to the heaven and help for the humankind. Thank you very much.
Thank you, Dr. Majumdar, for a very enriching and enlightening talk. In fact, he touched upon a very complex topic in a very simple manner so that the students can understand. And I think I'm sure all of us got a message that economic viability and affordability of important therapeutic proteins or other products is also very important. So he was trying to tell us how, you can use animals to produce that. And another important issue which he touched was that, transgenic animals is a big issue. So how you can do without affecting the germline. So I'm sure the students would have got this message. I just want to know from Dr. Majumdar if he has time for a few questions.
Yes, madam.
So students, you can please ask your questions in chat box and we'll take them. Or you can raise your hand. So students can ask their questions, because it's a rare opportunity that you have a scientist of the caliber of Dr. Majumdar here to address you. So please make use of this opportunity. I don't see any questions in the chat box. I don't know whether live audience has any questions.
Ma'am, we have one question here.
Please go ahead. Make use of this opportunity. Very rarely you get a chance to interact with scientists of his caliber. So.
Good morning, ma'am.
Yourself also.
Ma'am, actually, my question was, if the gene that we are taking and we are isolating and we are putting it in a transgenic mouse, will that have any efficacy towards the consumption of the milk that would be produced by the mammary gland of the transgenic mice, or it will be of the same, will there be any variation in the transfer process that will happen when we are putting the gene in the
I got your
transgenic mouse?
question. I got your questions. I will give you two answers for this, for knowledge. Number one.
I'm not able to hear anything.
Can you hear? Can you hear?
I can hear.
Yeah. Can they hear?
Yes, sir.
Yeah. So two answers of this question. Number one is such kind of animal is precious. One animal will be worth several lakhs of rupees. Do you understand that medicine?
Yes. Yes,
this
sir.
milk is not for consumption. Even 10 liters of milk can fetch a whole country for a day for major diseases. You don't want to consume as a milk. 40 रुपए में कहीं लीटर 50 रुपए में खरीद लेना. आपको जितना दूध चाहिए. Number 1, that's the answer. This animal is a industry. You don't eat the milk. Second, suppose you want to eat the milk, तो हम लोग क्या प्रोड्यूस कर रहे हैं पूर्वा? प्रोटीन कर रहे हैं ना? हाँ, तो जैसे ही बॉयल करेंगे खत्म हो जाएगा, पी लेना, प्रेसिपिटेट हो जाएगा, पनीर बना लीजिए, उसका क्या है, खत्म, मिल्क की स्ट्रक्चर ही खत्म हो जाएगी, बॉयल कर लीजिए, टूट जाएगा.
मतलब the question was basically like, will there be any variation कि मतलब वो जो प्रोटीन होगा, the protein that we are synthesizing, ठीक है,
हाँ
वो
हाँ,
प्रोटीन में कोई ऐसा कुछ आपने कुछ एफिकेसी या कोई वेरिएशन तो नहीं आएगा कि
मैंने आपको बायोलॉजिकल एक्टिविटी दिखाई एक प्रोटीन की, देखा ना आपने?
यस यस
अमाइन
सर
एसिड की आना ही नहीं चाहिए। मैमेलिया लोग बैक्टीरिया में बनाते हैं, है ना, और पिस्टिया पास्चोरिस में बनाते हैं, उससे तो बहुत अच्छा है मैमेलियन सिस्टम। पोस्ट ट्रांसलेशनल मॉडिफिकेशन पूरा होगा, वही सबसे उत्तम है। और दूध से उत्तम कुछ नहीं है। मैक्सिमम प्रोटीन्स आपको मिल्क में मिलते हैं और सीरम में मिलता है ना।
यस
यस,
यस थैंक यू सर, थैंक यू सो मच.
Good morning, sir. My name is Manya. I'm pursuing BSc Honors Zoology. My question was,
Okay.
no matter what we're doing, at least these are transgenic mice, and we are reconstructing their natural genetic material. So first of all, would it not be of something like an ethical perspective? And second of all, in the longer run, do we not consider that what mutations can it undergo and what would it result
No,
into?
that is the reason such kind of thoughts come into many people's mind. That is why we have found the alternative, that don't generate transgenic animal. Only inject in the udder. Udder gland injection is not transgenic animal. Transgenic animals means foreign gene is into the head, brain, neck, eye, testis, ovary, right? What are we doing? We are going from GMO to direct transcription of that. ठीक है?
Okay, sir. Thank you
जो
so
आप
much.
कह रहे हैं वही थॉट्स हमारे पास भी है.
Yes, sir.
Sure.
So if there are no more questions, I would like to thank Dr. Majumdar for your very enlightening talk.
Just a minute, ma'am. I thank you. I just wanted to thank Dr. Subir Mazumdar personally also, because in spite of a very busy schedule, he has joined us today. And also, I just had one request from sir, that sir, today the audience that we have, it's a multidisciplinary conference, and the audience that we have consists of both social sciences as well as sciences. So if you could, in 2 lines, give the crux of your talk for our students who are from social sciences, it would be very helpful for everybody.
Sure. So basically, there are so many diseases at today's date which poor people cannot afford. No way. Forget about poor. Even I may not afford. Because once I was in hospital with Professor Chandana Das, she was head of the Department of Biochemistry, All Indian Institute of Medical Science. She was admitted for a bone marrow disorder. And then doctor came in. So she asked me, Subir, रेफ्रिजरेटर से वायल निकाल के डॉक्टर को दे दो. डॉक्टर को वायल दे दिया तो उसने पर्दा खींच लिया और उसने वायल निकाल के ढक्कन मेरे को दे दिया और मैडम को बेसिकली हिप रीजन में इंजेक्शन दे रहा था. So I was sitting and reading, that's 13,000 rupees human gamma interferon. From there it came to my mind. Then when the doctor went, I said, मैडम ये 13,000 का है, कितना इंजेक्शन लगेगा? तो शी सेड की सुबीर 6 महीने तक का इंजेक्शन है, हर 2 हफ्ते में एक इंजेक्शन. तो बोले इतना पैसा? तो बोली नहीं नहीं, मैं तो एम्स की डॉक्टर रही हूँ, इसलिए मेरे को एम्स से ये लोग सप्लाई दे रहे हैं. मतलब वो रिक्शे वाले का, टांगे वाले का, नॉर्मल आदमियों का क्या होगा, इतना पैसा कहाँ से आएगा? बोले हाँ, वो तो ट्रीटमेंट नहीं कर सकते. So the whole thought came in that how to help poor people, how because these are all generated in industry, that too in foreign countries. तो इंडस्ट्री का अपना ही खर्चा इतना है कि उसका कॉस्ट इतना आएगा. डेफिनेटली आएगा, क्योंकि उसको बड़ी बिल्डिंग बनानी है, एयर कंडीशन रखना है, फर्मेंटर्स मेंटेन करने हैं, स्टेरिलाइजेशन मेंटेन करना है, बैक्टीरियल कल्चर्स करना है, साइंटिस्ट चाहिए, केमिस्ट चाहिए, 3 शिफ्ट में काम करने वाले वर्कर चाहिए, सफाई के कर्मचारी चाहिए, अलग अलग लाइसेंस लेनी है. सो उसके बदले अगर हम लोग ऐसा बायोरिएक्टर इसलिए कहते हैं कि दिस इज बायोलॉजिकल, बट रिएक्टर मीन्स इंडस्ट्री वेयर समथिंग इज प्रोड्यूस्ड रिपीटेडली. तो अगर अडर ग्लैंड ऑफ द एनिमल्स कैन बी यूज्ड, अगर उसमें हम लोग इस तरह की प्रोटीन प्रोड्यूस कर सकें, क्योंकि प्रोटीन तो आखिरकार शरीर का प्रोडक्ट है, ये तो हम सिंथेटिकली बाहर पैदा कर रहे थे. अगर वापस इसे शरीर में ले जाके दूध में ह्यूमन का प्रोटीन प्रोड्यूस कर सके, तो फिर प्रोटीन बनने के बाद उसको एक्सट्रैक्ट करके और बड़े सस्ते दामों में दूध मिलता है. दूध की कीमत तो ले दे के वही रहेगी, उससे इतनी महंगी प्रोटीन निकल सकती है, तो उसको हम this disorder of your bone marrow, and total expenditure for 6 months is 5,984 rupees. So probably he will sell some of his property and generate 5,000 rupee. Maybe he will sell out of 4 cows, 1 cow, and generate some money to get the treatment. Even then, it will become feasible. Or if his sons are now, आजकल तो पैसे वाले हो गए सब, 5-6 हजार रुपए कोई बहुत बड़ी बात नहीं है. So we are trying to reduce the cost of the production of the therapeutic proteins in our country.
Thank you, sir, for explaining it so well. And I'm sure it has made a lot of sense to everyone. The importance of this work that you have been pursuing for so long. And if you can we take one more question from a student?
Yeah, last question. Yeah, I have to go
Oh,
for the meeting.
yeah, I know, sir. I know, sir.
Yeah, sure.
Good morning, sir. I wanted to ask, will this have any negative effect on the animal? Like as a long-term process, is there
नहीं
any
नहीं
negative
नहीं
impact on the animal?
नहीं, बच्चा, हर माँ बच्चा प्रोड्यूस करती है ना, तो हर माँ दूध पिलाती है बच्चे को टन टन दूध में प्रोटीन मिलता है. वेरी ह्यूज कंसंट्रेशन ऑफ द प्रोटीन्स ऑफ बेबी गेट्स. दैट प्रोटीन डजंट लीक बैक टू मदर्स ब्लड बिकॉज इट्स एक्सोक्राइन, एक्सोजीनस डक्ट से मिल्क बाहर जा रहा है. वो बैक फायर नहीं करता है ब्लड में, ये वन वे ट्रैफिक है. तो जितना भी प्रोटीन बनाएंगे वो बाहर जाएगा. तो माँ के ब्लड में न तो बच्चे को दूध पिलाते हुए माँ के ब्लड में ऐसा नहीं कि सब बढ़ जाता है बीटा लैक्टोग्लोबुलिन वगैरह, कभी नहीं. It's exogenous gland. That's a safety. Don't worry.
Thank you, sir.
So rather, I thank all of you for patient listening, and it's nice to interact with all of you. And I thank Suman Goyal madam and the organizers. Thank you. Take care. Bye.
Bye.
Thank you, sir, for the wonderful talk, and thank you, ma'am. I now request our principal, Professor Haritma Chopra, ma'am, to address the gathering and for the concluding remarks. Very important thing. I was
Even when they have to do the registrations is done through the QR code only, because this is what has been observed by me when I look at the various posters that are being prepared. So the students are very much tech savvy and they understand. And in fact, our college has also signed a MOU with Special Olympic Bharat, where the specially challenged students, then we are giving them the fitness training also, so they come in the morning. In fact, now the center is available open 365 days. So this is how Maitreyi believes in, and this is how we practice. And I take this opportunity to invite Dr. Michelle to come here and speak with you and maybe share with you how things happen in their university and what different things that she is looking at. So I invite Dr. Michelle. Thank you so much. Sorry. Thank you very much, Madam, for inviting me so warmly to speak. And it's wonderful to be here. I also thank Dr. Smita Singh for inviting me here and for warmly welcoming me as well. So thank you for that. I'm an anthropologist, which is the equivalent of sociology here. And my research has mostly been on disability in India. And I'm very interested in how India has been accommodating and including and conceptualizing disability and how that has changed over the years. We often think about the West, and I'm saying this in quotes, the West being ahead and the East in quotes being behind. But I don't think that's true at all. And I think it's very important to look at the specific and local ways that people accommodate and work on and with people with disabilities. And so to me, I think it's really interesting to think about what inclusion looks like in an Indian context. What does accessibility look like in Indian context? And how do we even think about accessibility in general? And I also want to say, when I work in India, I talk with a lot of government ministers who say things like, in the future, we will eradicate disability. There will be no disability in the future. However, that's not true. We will always have disability in the world. And I think it's interesting and important to think about the ways that disability is a form of diversity, is a form of human diversity, and disability brings a unique perspective, and disability can be a source of strength, and it can be a source of gain. Often when we talk about disability, we talk about loss, hearing loss or hearing impairment, and we think about that in a negative sense. However, I think it's really important to think about the positive things that disability brings to the world, to think about futures in which disability is present and valued. And I think, Madam talked about the ways that we need to think about including all people and allowing all people to contribute to the nation. And so I think, again, we need to think about the unique perspectives that disability brings to the nation and disability inclusion bring. So in my research, I will be looking at enabling units across India in both public and private universities and thinking about what inclusion and accommodation look like in these settings. Previously, I conducted research with deaf Indian Sign Language speakers, and I looked at how deaf young adults did not want hearing aids, they did not want cochlear implants. What they wanted was more deaf signers, and they also wanted more hearing people to learn Indian Sign Language. And to them, Indian Sign Language was a beautiful language, a national language that more people, deaf and hearing, should use. And similarly, I looked at government programs providing below poverty line children with cochlear implants. For those of you who don't know, cochlear implants are incredibly sophisticated technology, and the government has been spending 6 lakhs per child to provide cochlear implants, and these work for some children, but not for all children. And families often told me that they were told not to use Indian Sign Language. They were told, they should only listen and speak and focus on speech with their children. And I think it's really important that families be exposed to all modalities, and we know so much about how Indian mothers and Indian families work so hard for their disabled children, and for them to not be told about Indian Sign Language as an option is really a problem. So in that case, what we saw was a focus on technological solution. We're going to provide this very expensive machine. We're going to provide this very expensive implant, but we're not going to think about the social factors, and we're not going to think about the fact that Indian Sign Language is something that children and families can use. And so I think it's really important that when we think about research and when we think about social transformation, that we think about social factors more broadly, that we're not just thinking about technology. So, and I think we see that in the enabling unit here, as Madam was talking, we see students working together to create change. And that change may be technology in the form of QR codes, but we actually see students working together, and we see the important role that the social plays. So thank you very much for so warmly welcoming me. Thank you. Thank you. Thank you so much. Do the students want to ask Dr. Michelle any questions, or do they want to interact? You can raise your hands. Anybody? I think this was a surprise thing that happened. They were not expecting right now. So, but we are really happy to have you, Dr. Michelle, and she is, as I could see that you are a medical anthropologist, and you've been working in the field of deafness and implants and aids, and she has been working in the Indian context a lot, and it can be seen from the attire that you are wearing that you have really embraced our Indian culture also. So a big round of applause, and thank you for being here with us. So now I would call Dr. Meena Yadav everybody is waiting for the last session. So about the prize distribution, we have had 46 presentations, and also today we are going to give the prizes for the upcoming issue of Vantage's cover page, which has been designed by students. So,
Thank you, Brother. Hello. Am I audible? Hello. Thank you, Bratati. The much awaited moment is here for all of you. We have had 2 days of intensive engagement with different themes to our understanding of research, development, and social transformation. We could see the hard work that has gone into the preparations and presentations by the students. All the presentations were marvelous. All the listeners, the audience enjoyed them, as well as the jury members, and they must have had a difficult time deciding upon the winners. So I'm sure that the conference has been an enriching experience to all of you. Just listening to the speakers must have been a learning experience to you all. We thank our external and internal jury members for taking out time from their busy schedules and being here with us to evaluate the presentations. And I'm sure their suggestions and their comments will certainly help the students to improve their work in future.
We have external jury members from different renowned institutes. So let me take the names of all our external jury members. Professor Sabyasachi from Department of Sociology, Jamia Millia Islamia. Professor Vaishali from IP University, Delhi. Dr. Bharti Singh from Delhi Technological University. Dr. Jogeshwar S. Purohit from Classical Innovation Center, DU. Professor Artri Srivastava from NIPA. Dr. Simran Lohan, which she has interacted with you today also, Senior Advisor, Department of Biotechnology, Government of India, and Professor Rakesh K. Parasar from the Chemistry Department, University of Delhi. So these are very, experienced, renowned jury members. Now, before we come to the main, winners of the poster and the oral presentations, as Dr. Bhattadi already said that we would be announcing the winners for the cover page of our October 2023 issue of Vantage Journal of Thematic Analysis. You must have gone through the Equinox website, and there here mentioned that you can submit your entries for the cover page. So we have received several entries, and few of the entries we would be displaying here today for you, and the editorial board has gone to each and every one of them, because along with the cover page we have also asked them to submit a write-up about the thought behind the cover page. So we have also received these from the for the submissions. So we have selected few cover pages. So can you please play? So this is first one. This is from Monica, BSc Program Life Science, third year. Ritika Kalra, this is student of BSc Biotechnology, Kerry Institute of Advanced Studies. She has been an undergraduate student, zoology honors student from our college only. Next. Nikita Regu, BA program. Krishna, BSc Botany Honors from Gargi College. She has submitted to Gunjan Kumari from Government Girls Senior Secondary School, Delhi. She just sent the cover page earlier. So we contacted her. Please send your affiliation. So she said she's from school. So we were very interested. They have got information about submitting this cover page. So it was really nice for her to submit. So these were few selected cover pages, and the editorial board we looked at each one of them, and we have decided the winner for the cover page, and which will be published along with the October 2023 issue of Vantage Journal of Thematic Analysis. So the winner is winner is Ms. Ritika Kalra. Ritika, if you're here, we would like to. Are you here? No, she's not here. So this is the cover page which we are going to use for our upcoming issue of Vantage. So we have a very beautiful write-up also for this, which we will be sharing with you all on our social media handles once this issue is published. So for the winner, as we have already mentioned, she will be receiving a prize of rupees 1,500 for her contribution as a prize money, which will be credited to her account later on. We'll collect the details from her. So after this, now I would invite Dr. Bhakti Roy to announce the winners of the best questions, because from day one we have been telling you all that please ask the questions to the speakers, listen to the talks carefully, and there are prizes for it. So we'll be announcing a few prizes here. So at Mathri College, we encourage the spirit of questioning of interrogating of idea and the zeal to know more. In this spirit, for all events that are organized by the Center for Research, we always give out a prize for the best question that has been asked during the session. So today we have 5 prizes for different sessions. The first, I'll call upon Manya Sharma from Zoology Department. I request Principal Ma'am to please उधर मैम के पास जाओ. So it's a beautiful mug with Center for Research inscribed on it. Manya, if you could show the mug also. The next prize goes to Meenakshi Lakra of the Physics Department.
Can you read out the question that he asked during the session?
In fact, Manya has got the prize for asking a question in the session of Professor Aman Naban, wherein she asked about social conflict. So it's actually it's really appreciated that she's studying in the zoology department, but she asked a question in a session that was for the sociology department. Next, Meenakshi Lakra asked a question in the, I think, from Panjan sir's talk on yeah, you have to excuse me because I was not present in most of the session. I was just running about.
Let the winners inform everybody.
Okay.
What was your experience and what inspired you to ask that question?
Actually, I had no idea about Kanchan sir's presentation. I was like told at the last moment, but it was like the most informative and engaging presentation that I attended. And I really liked the topic because I had biotechnology in 11th and 12th, so I could really relate to his research work. And I was really happy to see such intense research being done. It was done in India actually. I was aware that gene chips and such things were in America and outside countries, but I was like really happy that sensing kits and chips were being developed in India, and like really interested. Maybe one day I could even work with the nanomaterials he was talking about.
That's very good. That should be the spirit. Very good.
Thank you, Meenakshi. You just saved my skin. Yes, ma'am. Yes, ma'am. I really like this one as well. Like, मेरा time जो छूटा है, like I'm like physics में अपना सब कुछ हो गया, and biotechnology I was so happy. Like, yeah, I'm relating to this. जब हम पढ़ रहे थे 11th 12th में, this is all future scope की future में कर सकते हैं. But he was like हम कर रहे हैं. So I'm like so happy to see all of this. Thank you, Meenakshi. And the next best question award goes to Kashish Mishra from the Physics Department again. So Physics Department was very active, it seems. In fact, I should mention here that while we were trying to find internal jury members, we realized that all the teachers in the physics department and chemistry department were involved in some of the other projects. Good morning, everyone. Myself Kashish from Physics Honors. मेरा जो एक्सपीरियंस था इक्विनॉक्स का वो बहुत ही अच्छा था क्योंकि फर्स्ट टाइम मैंने कोई रिसर्च करा और उसको प्रेजेंट किया. रिसर्च वर्क में भी मुझे बहुत अच्छा लगा और मुझे बहुत खुशी है कि मुझे इतना अच्छा प्लेटफॉर्म मिला अपना को एक्सप्रेस करने के लिए. मेरी कम्युनिकेशन स्किल्स भी बहुत अच्छी हुई. मैं इतना बोलती ही नहीं हूँ.
क्वेश्चन क्या पूछा?
मैम क्वेश्चन ये था कि उन्होंने बताया था कि ये सेंसर सेल्स होते हैं चिप्स में सॉरी सेंसर सेल्स होते हैं तो फिर मैं बताया था कि डिजीजेस को डिटेक्ट करने के लिए सेंसर्स यूज कर सकते हैं हम तो वो बहुत अच्छा है टाइम टेकिंग नहीं है और ज़्यादा एक्सपेंसिव नहीं है तो मैंने उनसे पूछा था कि क्या हमें मतलब ये सेंसर जल्दी डैमेज हो सकते हैं मतलब कि हमें ये जल्दी डैमेज हो सकते हैं और हम इसको कैसे प्रिवेंट कर सकते हैं अपने लेवल पे Thank you, Kashish. Thanks a lot. And I think the fact that you came here and you speak, it is the biggest achievement of the conference. So a big round of applause for Kashish. And the next award for today's session on water from air goes to Parul Mishra from the Sociology Department. And I'm sure many of you must have watched many of the sessions. We are going to upload all the recorded sessions in the Equinox website and also on the YouTube channel of Center for Research. Good morning, everyone. I'm Pari Mishra and I'm from the Sociology Department. And I actually very much enjoyed the research what Sir said because, you know, we are always facing this water scarcity problem everywhere. And as he mentioned, there are over 60 places which lack the amount of water required. So when he mentioned that you can acquire water from the atmosphere, I was very intrigued. And other than that, he told that they are working on such technology, which made me like very proud that India is, you know, developing so much. So that's it. Thank you. Thank you so much, Pari. And I would also like to inform all the students here that many of these talks, many of these research papers would be there in the coming issue of Vantage. Please go to the college website and go to the Vantage website where you will find many earlier issues also and very relevant themes where articles have been published. And the last prize for today goes to Dishita, third year, Life Science, who asked a question to Dr. Sabir Mazumdar, I think about health self-attack. It was a very nice presentation that Sir gave, but according to me, I wanted to know what were the negative impacts of the research because we know that every project has one thing or the other which is negative, which also leads to the future perspective, which led me to ask that question. Thank you, Dashika. Thank you so much, all of you. And I'll hand over to Dr. Mona Abov to continue with the session. I'm going to announce the winners, but before that, I would like to invite all the conveners on stage with peaceful manner so that you know every winner will be called on stage and we can have a good photograph. Please. So now we are coming to the most awaited moment of the conference. We have had 5 oral sessions and 1 poster session. While calculating the results, we have combined club sessions 3 and 4. So there will be only 1 winner for session 3 and 4 and 1 winner each for session 1, session 2 and session 5. And for poster presentations, there will be 2 winners, the first prize and the second prize. So to encourage the students, we are giving prize money also to them. The first prize is of rupees 2,000 and the second prize is rupees 1,500 and all these they will be credited to the accounts of the students. So while I am announcing the results one by one, I would request the winners to come on stage and have photographs with the conveners. So for session 1, that is, I will start with the oral presentation session. So technical session 1, the winner is Hera Saud from Department of Sociology, Jamia Millia Islamia, New Delhi. Her topic of presentation was critical theory and social transformation, reflections on the empowerment of the subject. Is Hera here? Okay. Now, coming to session 2, the winner of this session goes to the team that worked on on-site assessment of water and soil health for integrated social and environmental awareness among rural population. The authors of this presentation are Riti, Anjali, this is from chemistry department, Riti, Anjali, Mahima, Panchali, Sneha, Nyasa, Alima, Dr. Ghazala Rohi, Dr. Pinky Bajaj Gandhi, and Professor Haritma Chopra. So today you will be getting only a photograph, and e-certificates and cash prize will be credited into your account, right? Thank you, everyone. Now, for technical session 3 and 4, they were judged by the same jury, internal as well as external. So we have one winner from that session. The title of the winning presentation is Exploring Spatial Variation in Antioxidant Profile of Medicinal Plant Piper Langell. This is from the Department of Botany. And the authors are Sana Ahmed, Dikshita, Somin, Dr. Shweta Sharma, and Dr. Rama Sisodia.
Mentors can also.
दोनों नहीं श्वेता
Thank you, girls, and congratulations. Now, session 5. The winner is from Department of Commerce. The paper title is Revealing the Relevance of Influencer Marketing in the Food and Beverage Industry for Indian Generation Z Consumers. So authors are Akanksha, Priyambada, Radhika, Vanshika, Dr. Jyotsna, and Ms. Karishma Arora. Thank you, Jyotsna. So we are done with oral presentation, and poster presentations. We have 2 winners. The first winner is from Sri Venkateshwara College, an insight into vegetable adulteration, unveiling the deceptive practices. The authors are Akanksha Sharma, Anushi Singh, Itika Garg, Haya Deepti, Dr. Rajendra Fartar, and Dr. Anita Verma. Is anybody from Venkateshwara College here from this team? No? Anyways, congratulations. And the second prize goes to the paper from the Department of Botany again, analyzing the effect of Indian river water on phytochemical constituents of 3 species of Osamum, Authors are Preeti, Prachi, Sneha, Dr. Pooja Baweja, Dr. Pinky B. Gandhi, and Dr. Ankita Sehrawat. Can we have you on stage, please? Congratulations to all the winners. So many congratulations to all of you. Those who are here, they really enjoyed coming on the stage, and those who are not here, they'll be still. Anyways, our congratulations to all of you. Before we move to the formal vote of thanks, I would like the students again, would you like to give your feedback? Do you want to come here and say something about the 3 days of the conference that you were engaged with? Any student, please raise your hand. You can and come here. Any girl? Have you seen the feedback from all of you? Fill the feedback form. So nobody wants to speak here. You'll write there in the feedback form. Okay, thank you so much. Now I would invite Dr. Pinky Bajaj Gandhi to deliver a vote of thanks. Ma'am.
Thank you, Meena. Namaskar and a very good afternoon now. It has become afternoon. So, namaskar and very good afternoon to one and all. Now, it's after a very hectic schedule of 2.5 days, we have come to the conclusion ceremony of our this conference, Equinox, which is organized under the aegis of Vantage, the Journal of Thematic Analysis, and IQAC Material College. So this is the conclusion ceremony. So I would like to thank our chief guest for the ceremony, Professor Dr. Subiris Majumdar, for his contribution and for his prizing the audience with his talk on the uses of biotechnology. I would also like to thank Dr. Suman Govil for chairing the session and also always be there for the journal Vantage as a member of editorial board. Now, during the conference, there were 10 eminent invited speakers and various, which talked about in the various fields of social and science fields. And there were many participants who participated. There were a total of 35 oral presentations and 11 poster presentations showcased by the faculty, research scholars, and even the undergraduate students. And though this conference is of 3 days, so we have participated here only for 3 days, but it takes a preparation of many months, almost a year, for the conceptualization and execution of this conference. Because the first step starts from, it's a theme-based conference. The first step starts from choosing a theme, choosing an apt theme, and then which should be apt for all the fields, that is the social sciences or sciences. So first step is to choose the theme, and I would like to thank our advisory board, our editorial board of the Vantage for helping us to choose the theme which is apt for all the courses, that is the sciences and the social sciences. Now, through the lectures and the oral presentations in this conference, uh, we saw that we realized that science plays and the research plays a very important role in the social transformations made by the formation of a vaccine by an eminent scientist, or the young scientists, the young undergraduate researchers choosing formula or the remediation for polluted water, or creating the microbial cells which use bacteria for the generation of electricity. So this research plays a very, very important role in social transformations. Now, I would like to thank all the conveners of the conference and our young and energetic organizing team, because it's a very long team. Many members are there, but they work day and night to make this conference successful. Because without their help, the conference is, arranging these kinds of conferences is not possible. So I want you to give a round of applause for our organizing team, which have worked so hard, right from sending invites, then calling people, calling the invited speakers, then making the certificates, distribution of the certificates, all the work they do without any hiccups. So very many thanks to our organizing team. I would also like to thank our college administrative people, because they help us in all the logistics and the technical support. Because technical, whatever we require, whatever logistics are required, the administrative staff helps us in that. And last not the least, I would like to thank the chief editor of the journal Vantage and the principal of our college, Professor Haritma Chopra, for her vision and support for. She's always there for her, for anything we want, anything, and she's the chief editor also. So, thank you, ma'am. And this conference is an annual conference, and every time we have a different theme. So next year we will be back again with another new theme, and we anticipate your participation with new zeal and enthusiasm. Thank you. Thank you, Pinky ma'am, for your vote of thanks. And now I invite all the organizing committee team for. Before that, let us all rise up for the national anthem, and then we can have a group photograph. Janagan.
energy was slightly low. So when we are leaving this, before that, hip hip. 3 cheers for Metroid, Metroid teachers, Metroid students, Metroid family, Metroid kutum. So hip hip, hooray! Hip hip, hip hip. हाँ अब ठीक है, अब जा सकते हो. Organizing committee team, the very dynamic organizing committee team, to please come on the stage for a photograph.